Well, thank you everyone for joining. Like Peter said, I especially want to thank Bryn Dentinger, who is joining us tonight. He is originally from Minnesota. And I'm just going to tell you a little bit about his background. He is currently the curator of mycology at the Natural History Museum of Utah and an associate professor in the biology department at the University of Utah. Specifically, he runs the Dentinger Lab, which supports a wide array of research, including graduate, undergraduate, National Science Foundation funded research projects, investigating fungal biodiversity and evolution. Bryn hails from Minnesota, like I said, and received his PhD from the University of Minnesota, where he studied the molecular systematics of clavarioid and porcini mushrooms. He's carried out fieldwork all over the world, including exciting trips to Vietnam, Brazil, and Cameroon. Outside of work, he enjoys cooking, especially with mushrooms, and brewing beer. I hear he also likes whiskey, which is something I also really like. But that shows that even his hobbies often include a mycological component. We were asking him right before the meeting about the mushroom season in Utah, and Peter was saying he might have been there at the same time and been able to go hunting with them, and he said, no, I was probably in Minnesota at that time. So hopefully he can play his cards right and hit the mushroom hunting seasons in both places. But Dr. Dentinger, thank you so much for joining us tonight. We really appreciate it.
Thank you very much. Hello, everybody. Thanks for coming. I'm really honored and flattered to be invited to give a talk to the MMS, where I cut my mycological teeth quite a few years ago now. So thank you very much. And I was looking forward to the opportunity of upgrading my t-shirt, which I still wear, but it's about 20 years old now. So I was asked to talk about two really different topics tonight. So I've kind of split it not half and half, and that's because I don't have as much to talk about on the second one, and I'll explain why that is. But the overarching theme for the research that I do and my students do is really based on this idea that a lot of fungi, perhaps the majority, are engaged in a variety of symbioses with other organisms. And these are just a few examples, prominent examples of some antagonistic symbioses, where we have some plant pathogens on the left, and some insect pathogens, and even some mushroom pathogens. And then on the right-hand side, you'll see that there are some mutualistic symbioses, the best known perhaps being mycorrhizae, and of course, the two independently evolved systems of cultivation where termites and ants have both adopted fungiculture as their sole source of nutrition. And then lichens, I think, perhaps epitomize fungal symbioses, and that's illustrated in the middle. But what this is really trying to capture is just how ubiquitous symbioses are in fungal biology. And the questions that I'm really interested in trying to understand with the research is really how symbioses have shaped and impacted fungal biodiversity. So questions like, how do species evolve with other species? What is the genetic and functional basis of these associations? And what factors contribute to speciation in symbioses at varying scales of organization? And to do this, I have looked at a number of different systems, from diffuse symbioses, such as the mushroom mimicking Dracula orchids, where the orchids look like a mushroom and smell like a mushroom to attract pollinators, but this really has no impact on the reproductive success of the mushrooms that they co-occur with. And apologies, the image isn't showing up for some reason. To ectomycorrhizal mutualism, which there's a more intimate association where both partners really require each other for survival, but their reproductive cycles are independent and unlinked. To the fungus growing ant mutualism, which is an example of a strict mutualism, a strict symbiosis, where both the ants and the fungus rely on each other for reproduction. So today I'm going to just talk about the example of the ectomycorrhizal mutualism first, and then I will switch over and talk a little bit about the fungus growing ant mutualism. So sort of a context for starting to investigate these kinds of questions is establishing a fundamental baseline of who the organisms are And you probably are aware that we know most of the mammals in the world. We know most of the plants in the world, but this is not the case for fungi. So estimates range from 2 to 12 million species. That's a big range, really hard to get an estimate on what you don't know about. But we've described only 148,000 of them. No matter what the estimate is, there's a lot more than we've described. And so there's still a lot of just basic documentation that needs to be done. And so a lot of my work involves just trying to document this diversity. And I do this through a variety of different ways. One, of course, is doing a lot of field work around the world, targeting areas that are underexplored, which can include your backyard. Engaging with citizen scientists. So as many of you are well aware, I'm sure, finding a fungus is a happy accident in most cases. And so if you really want to be able to document things fully, you need to have a large army of people that are out there all the time. And engaging with citizen scientists is really an excellent way. And I'll show you some examples of how important this has been to my own research. And then also finding new diversity in perhaps some unexpected places like local food markets whenever I'm traveling, and even supermarkets. And I'll tell you a story about that. And then finally, of course, it's hard to overstate the importance of museum collections and fungaria as a resource for documenting fungal diversity. So just as an example, this is one of the recent projects that I've been doing with a couple of collaborators in Cameroon, where we set up these 1 hectare plots in this lowland forest and surveyed them intensively and systematically over a period of 4 years. And as an example here, of the 700 or so collections that we made, we found 112 species of Boletaceae in these 3 hectare plots. So there's quite a lot of diversity there. You can see some of the remarkable things that we found that we're still trying to work out. And I'll show you some preliminary results today of some phylogenetic placement of these species. Here's some new species of Porcini, and these belong to the strict Porcini clade, Boletus sensu stricto. These are new, all of these are undescribed species that have been contributed to our lab through citizen scientists. So everything from that Smurf-like blue Porcini, that remarkable thing on the left from Taiwan, to the one on the bottom here comes from Texas. And this one actually has a really important evolutionary placement that I'll talk about in a minute. And then I did allude to the fact that you might find new diversity in somewhat unexpected places, so your backyard perhaps, but even the grocery store. And this is an example of some species of Porcini that we found by going and buying a packet of dried Porcini that was being sold as food. And we pulled out 15 fragments from this packet and sequenced the DNA barcode region. And when we did that, we found that there were 3 clusters of those sequences that you can see on the figure on the right. There's the phylogeny, and then there's the multiple sequence alignment of the barcode region next to it. And you can see quite clearly that there's differences between those groups. Now we named them as new because they didn't have names that we could use. Now you can see from the spores that these are indistinguishable microscopically. So we wouldn't have been able to tell just using a microscope from that packet what was in there, how many species were in there. So it really required us to use these DNA barcoding methods to find them. We followed up on this. I had a really excellent undergraduate student who came and did an independent research project in my lab. He decided to pursue this even further, and he bought a bunch of food products, both at our local grocery stores and then from a large online retailer who shall go unnamed, but you might be able to guess who that is. And then we did meta barcoding. So we amplified the ITS region from all of these food products. And in many cases, of course, there's a mixture of species. And so to sort those out, we had to use high throughput sequencing technology. And so we used Illumina sequencing to sort out the different sequences in them. And we found some surprising results. The general result we found was that most of these food products were not accurately providing information about what kinds of mushrooms were in the products. often they were mostly Swillus or Agaricus, Bisporus, or Shiitake when they were listing truly wild mushrooms on the labels, or things like Porcini and substituting Swillus for it. But there was one case that we found rather disturbing, and that is the picture here in the middle. This is the contents of that packet, which is sample D in the upper right corner of the panel. And the arrows are pointing to 3 pieces in that packet that you can see quite clearly are from lamellate mushrooms, whereas the package clearly states that these are supposed to be Porcini. And when we identified the contents, we found 7 species. We could identify 7 species of mushroom, which are listed below. And you'll probably see right away that none of those are Porcini. That Boletaceae is an unidentified species. We couldn't put a name on it. And then that last species might be somewhat alarming, Amanita pseudoporphyria. And indeed, that species has been implicated in causing acute renal failure in humans. And perhaps not surprisingly, if you go to the product page for this packet, which was purchased from an online retailer, there are a number of testimonials that suggest that probably you shouldn't be eating these things. And I'll let you take a glance at some of those testimonials. So we notified this retailer in as of the publication of our article, which was about a year after the product was still available for sale, and at one point was even being promoted. So new diversity, even in unexpected places. But of course, the Fungaria are really a place that we can continue to investigate and interrogate, especially with these new molecular methods that we have. And one of the really important things, I think, is the fact that these are really, if you want diversity, the best place to go is at a fungarium, because they represent an accumulation of effort over many years and by many people. And so they are the most diverse places on the planet when it comes to fungal diversity. Now, of course, none of that's living, but the reason that these specimens are of really high value is because, generally speaking, there just aren't very many people collecting fungi relative to other groups of organisms. The fungi, of course, are ephemeral, so they disappear quickly, and so that just makes them that much harder to document. They're difficult to preserve, so they will spoil rather quickly if you don't have adequate means of drying them. And then as an illustration of just how diverse and important Fungaria are, this is just a kind of quick statistic about what was at Kew when I was there. We had 150,000 specimens databased. So this represents about 10% of the specimens at Kew. And about 80% of those were represented by less than 10 specimens each. And 40% of them were represented by a single specimen. So Fungaria tend to have a lot of diversity, but not a lot of depth. Okay, so we really, I think, need to make as much use of the existing collections as possible to fill this diversity gap. That's where we should be focusing a lot of effort. But the problem has been that they're really difficult. And that's because sometimes they're small. Like this is the holotype specimen of Boletus leptocephalus. That's all that exists of it. So there's not a lot to work with to begin at the beginning. And then if you extract DNA from it, if you can achieve a DNA extraction from a sample like this, and sometimes you cannot, the DNA tends to be really fragmented. And so this is a gel showing you the sizes of DNA fragments. And you can see that most of these are down at this low level. And in fact, other studies have shown that older barium DNA, and this is true for Fungarium DNA as well, is as highly fragmented as you would find in an animal remain of thousands of years ago. So this is really, we're really dealing with an ancient DNA-like problem when we want to go in and take advantage of these older collections. Now, of course, what's really important about the old collections in many cases is they are the names that we're trying to apply to modern taxa. And if we can't understand what they are, then how are we ever going to name the things that we find today? There's a couple of other issues in historical specimens that are more technical in nature. So the fragmentation process itself is the big source of that fragmentation pattern that you see in a gel. And this is through spontaneous hydrolysis. And the other problem is this deamination. And this is a this is a feature of ancient DNA samples, something you'd find in Neanderthal DNA or mammoth DNA. And it's unclear whether or not this is an issue in old Fungarium specimens. And this is where you have hydrolysis of the amine group on a cytosine residue. And if you don't remember your high school biology, DNA is made up of 4 nucleotides: adenine, guanine, cytosine, and thymine. So this is one of the nucleotides. And what happens is when that amine group, which is the NH2, gets cut off, it gets turned into a uracil. And a uracil is the same as a thymine residue in an RNA molecule. And so what happens is it looks like you get a cytosine to thymine bias in the ancient DNA samples. The other problem, and this is perhaps the biggest problem we face, is that very little of the DNA that's present in these old specimens are actually from the target organism. The vast majority come from contaminant organisms, both people that have handled the specimen or molds perhaps that have grown on the specimen, bacteria and other things. So this is another big problem. Okay, so we set out to try to see if we could get DNA preps and sequence them from really old types, such as this is the possible holotype collection of Boletus edulis var clavipes Peck from around 1889. And we were successful in extracting DNA, and you can see the plot on the right shows that the average fragment size was only 58 base pairs long. Now, this is too short to efficiently use PCR to access, and so we have to use other technologies. And so we attempted to use Illumina sequencing. And one thing, I sort of glanced over this because I didn't think it was going to be that interesting to you, but we can look at both double-stranded DNA, so DNA as you would normally find it in your cell, and single-stranded DNA by doing a different sort of preparation of the DNA prior to sequencing. And the reason that we might look at single-stranded DNA instead of double-stranded DNA is because the single-stranded DNA has been shown to have a higher endogenous signal. More of it comes from the target organism. And there are a couple of other features that may make it advantageous. Now, this is mostly known from ancient DNA samples and not known necessarily from these less ancient samples from the Fungarium. But in any case, we tested both. And first, I want to show you that deamination that I was talking about, where you get the cytosine turning into a thymine, that actually happens at a fairly high rate in the Fungarium specimens that we looked at. So this is an example from the holotype of Boletus frustulosus, which was collected in 1897. And you can see that as at the ends of the graphs, the red line goes up. And that is that deamination problem that happens at the ends of the fragments. And so we were getting about a 5% conversion of cytosines to thymines at the ends of the DNA fragments. Okay, then we wanted to look at how much of the DNA comes from the target organism. And the good news is we could find fungal DNA. We're using a special method to try to identify what organism the sequence comes from. I won't go into detail unless you're interested. But of course, we found other fungi in there that we didn't want. And here's some examples: Aspergillus and Trichophyton. We also found bacteria, not surprisingly, of a variety of types. We also found some animals. So chordates, this may be human DNA, it may be mouse DNA or other vertebrate DNA. And then also we found some arthropods, and in this one, Lasioderma comes from a well-known herbarium pest. And then there's a bunch of other stuff that we couldn't identify, that's the white dots. And then the vast majority is unclassifiable. And you can see that there's 2 separate clouds of these unclassifiable dots. A lot of the unclassifiable stuff at the top is likely to come from the target organism, actually. And that's because there's just not a reference genome. So our ability to annotate the sequences isn't very good. The stuff on the bottom, that is all contaminant DNA and should be removed for downstream processing. Now, that was double-stranded DNA. When we looked at the single-stranded DNA, it performed as predicted, where we actually got far less contaminant DNA. So most of that DNA looks like it's probably from the target organism. Okay. Now, I'm skipping over a bunch of stuff, but basically we sequenced a number of types and old specimens. The oldest one dated back to 1849 and was collected by Hooker in Sikkim, India. And this is the type of Boletus gigas. So we took the ITS sequences that we got out of the genomes and we put them into a phylogenetic tree of all Boletaceae species that are publicly available, which is that giant mess of lines on the left. So I just am highlighting a few of the examples here of what we found. So it turns out Boletus gigas, that specimen from 1849, we were able to show is actually a Lexinum. So its name will now have to be changed to Lexinum gigas. Boletus multipunctus Peck, which has been suggested to be a member of the Porcini group, that turns out to not be a member of Boletus sensu stricto. So it ends up being somewhere else in the Boletaceae. Boletus nobilissimus, which is the species that that is common in Minnesota, that this is the name that I've been giving it since I was in graduate school. It is identical to the type of Boletus nobilissimus. It turns out that Peck named it way back in I can't remember, early 20th century, let's say. And it's Boletus variopes. So this is a species that has been misinterpreted by many people for a very long time. And it turns out that what we call Boletus nobilissimus today is really what Peck was calling Boletus variopes. Boletus edulis var clavipes is a problem, and that's because the only sequence we can get from it is identical to the type of Boletus atkinsonii. Now, it's unclear whether the collection we sequenced is the true holotype because Peck never referred to a specific collection. And so we don't quite know how to explain what's going on with the edulis var clavipes, if that was really truly something that has been misinterpreted for many years, or if Peck may have just been mixing collections and we just happened to sample one of the Atkinsonii collection specimens in the collection with clavipes. Peck was known to do that kind of thing. So we don't know what the answer to that is yet. Boletus madiae has been an enigma for a very long time. It is known by a single collection, the holotype collection that Harry Tiers designated. The specimen came from the Sierra Nevada range, and no one's really known how to interpret it today. And it turns out that I can confidently say that the sequence I generated as a graduate student in Dave's lab was correct. And it turns out that this is identical to what has been named recently as Boletus regenius. So Boletus regenius will have to change names unless there's a movement to conserve the name, because it is actually the same thing as Boletus madiae. And then finally, something that I think a lot of people will not be too surprised by, people that are familiar with these species, but Boletus gertrudiae, which is a species similar to Boletus separans, but was designated as different by Peck because it had a yellow ring at the top of the stipe. It turns out that this is conspecific with Boletus nobilis Peck. So these are just some of the discoveries we've made by being able to access genome sequences from these old Peck types, Peck and other type specimens. So we now have 32 species of the Porcini clade that are represented by type sequences, sequences from type specimens. It turns out that there's 13 species in this clade that still do not have scientific names. These are new species. 4 of these do not have physical types. That's the European taxa, because they were named from paintings. One is a so-called dark taxon, meaning it's known only from a root tip. And then I've been able to fix 8 names in this phylogeny, 3 of which have not been applied in over 100 years, and some of which are going to be predate some of the newer names. So this is all unpublished, but we hopefully we'll get around to getting this published this year. Okay, I thought you might be interested in knowing where some of these new species landed on the tree. So here's 6 new species from Asia. And you can see that one of them is kind of an early diverging lineage. That makes it quite interesting phylogenetically. And one of these I wanted to point out is a contribution from a citizen scientist. These 5 are from North America. You might be surprised to learn that, yes, even in North America, there are still undescribed species of Porcini walking around outside. 3 of these are from citizen scientist contributions. Now, the one on the bottom, I mentioned briefly, but I wanted to point out the fact that this belongs right here. This is a very old lineage of Porcini. So quite surprising to me that this had not been known and is occupying this really old position within the phylogeny of Porcini. I should say that this topology is not correct. We now have a correct topology from whole genomes. This is just an ITS-based tree. So it turns out that this one right here is one of the oldest, earliest diverging lineages, and it's in Texas, perhaps elsewhere. Okay. So I just spent quite a while talking about diversity and how to document diversity. So I want to shift gears now a little bit and talk about more on, what use can we make out of this diversity once it's documented. And as a case study, of course, I'm going to talk about Porcini mushrooms, the Boletaceae or boletes. And these are really mushrooms with tubes. And they are globally distributed. The reason that I think it's a good group to look at is because of their global distribution. They're a very species-rich clade. There's over 1,000 species known. And I know that there's many more out there. I think Porcini is a good example of how many are still lurking out there. It's a very morphologically diverse group. You can see some examples of that. There's a number of truffles that have evolved in the group. Nearly all obligately ectomycorrhizal and known from all ectomycorrhizal habitats surveyed, or nearly all, I should say. And in those habitats, they're almost always among the top 3 most diverse and abundant groups. So they're important ecologically. They also display a really broad ecological amplitude. You can find them in at high elevation, at sea level, all over the place. So they represent a great deal of ecological diversity. And then, of course, it would be remiss I don't want to be remiss to not mention that they're economically valuable. Of course, edible Porcini, being one of the most widely traded wild edible mushrooms in the world, is estimated to be worth over $100 million US dollars annually. Okay, so traditionally, the Boletaceae were classified by morphological features, such as basidio-spore morphology, trauma anatomy, and specialized cells like the cystidia, as well as secondary metabolite chemistry, such as pigments. But that hasn't resolved a lot of the relationships. So today, molecular phylogenetics dominates Boletaceae classification, as it does in other groups of fungi. Yet, despite a lot of effort over the years, although the trees get bigger and more colorful, it turns out that the backbone relationship, so the relationship amongst all of these lineages, has never been resolved. And this is a quote from one of my PhD committee members. He said that this was an insurmountable obstacle, and I'm here to prove him wrong, finally. It's taken me a while, but the backbone of the tree being unresolved means that you can't test topological hypotheses, such as biogeography or trait evolution, things that we want to use a phylogeny for. If a phylogeny is going to be useful, it's going to be useful to predict things. But if you don't know the relationships, then you can't predict anything with any certainty. And what's happening here is the Boletaceae underwent an evolutionary radiation. So they are the birds of the mushrooms. They diversified very rapidly. And that means there's very little recorded ancestry in their genomes. This is also a recipe for something known as incomplete lineage sorting, but you don't need to worry about that. As a result, the boundaries between these clades has made it so that people have been describing each clade as its own genus. This has been a trend over the last few years. This is actually a bit out of date, but you can see there's been a rapid rise in the number of genera that are being used to name these groups, even though the backbone, the relationships between them, is not established. So this is resulting in a lot of monotypic genera. So the logical conclusion of what's been happening is that every species of Boletaceae is going to be its own genus now, which I don't find very helpful. So I attempted to try to find a solution to this problem, which is a lot more data, but also more representative taxon sampling. And by this, I mean being more comprehensive with sampling the known diversity based on taxonomic classification, but also geographic diversity. There have been large regions of the world that have not been sampled. And the rationale for using whole genome sequencing is that it's actually really kind of cheap to do in fungi. And that's because their genomes are on average quite a bit smaller than you find in plants and animals. And this is just an example. The average is 43 mega base pairs. That's million base pairs long. In comparison, a human is 3 billion base pairs long. So it's actually really cheap for us to do this kind of data generation. So what we did, and wow, that looks really terrible. Sorry. I don't know what happened there, but what we did is we've sequenced 245 specimens of Boletaceae, and this is kind of the geographic breakdown of those specimens. We went to great pains to include things from regions that were poorly represented in prior data sets. This includes Africa and Madagascar, where essentially there have been none included in the past. These are collections that I have made through work in Cameroon and Madagascar. And then also a lot from Southeast Asia, where there's a lot of diversity in this family in that area. And what you can't see is I just said, basically, we sequenced them with Illumina sequencing. So this is what we got. This is the current tree. So we were able to use about 1,300 genes. And the good news is every node in this tree has full statistical support or nearly full statistical support. So Bob Zink, if you're out there, although I don't think you are, it's not an insurmountable problem. I've finally solved it. And I just wanted to show you one part of this tree. This is the Porcini clade. So that's what this is here. And you can see, if you can see my cursor, this is that new species from Texas. So you can see just how important evolutionarily that species is in understanding the origins of Porcini. Without that, you might have inferred from these two species that the origin of the group was Southeast Asia. This puts that into question. Okay, so there it is, a reminder. The other thing I think is really fun for me is trying to figure out where Porcini came from, not just geographically, but what else they're related to. And it turns out that the closest lineage now is this one right here. And this is a species from Africa. And that's it pictured right there. And this species is curious because, as you might infer from the specific epithet, it smells like raw garlic. But it has a lot of features that reminded me at the time of Porcini, and I even made notes about it when I was describing it in the field. And so I think with this understanding, we might finally be able to reconstruct what an ancestral Porcini looked like, what the features were that was present in the common ancestor. So to me, this is a very exciting find. Okay. So we also are looking at, we're interested in the diversity that forms even below the species level. And as many of you might have experienced in the field, Boletus edulis is a morphospecies complex. These pictures are all Boletus edulis from different places, including Minnesota, which are the pale one here, and the orange one here. These are from Minnesota. These are from Europe, and this is from southern Colorado. So what's really interesting is this species is really widespread. It's found throughout the Northern Hemisphere. Just like the family, it exhibits this really large ecological amplitudes. You can find it at sea level and then all the way up to subalpine and tundra habitats. And then it's also associated with every known ectomycorrhizal host in these regions. although there is some difference in the distribution of that association. And then it's also been introduced a number of times in its non-native range. And that gives us an opportunity to really understand aspects of its population dynamics that we couldn't by observing only the native range. So you might be wondering, are we looking at one or more species? And this is a question that I've been trying to answer and still do not have an answer for quite a long time. And the problem here is the ITS region, the barcode region, is not sufficient. So you can see that there's some implication of some geographic structuring based on the colors of the lines, but that is not held up when you compare it with another gene like EF1 alpha. So what we're looking at here, we think, is a species that's in the process of diverging. It's in the gray zone between a single species and two divergent species. So it's at some point in that zone of differentiation. And that makes it a very interesting study organism, because we can learn something about the speciation process by looking at it closely. So one thing we're trying to understand is some of the basic evolutionary and demographic processes that are affecting the population structure in this species. And as an example, we don't really even know how dispersal contributes to the establishment and distribution of most EM fungi. So we know that these spores are tiny and can travel long distances potentially on the wind, but we don't know how much that actually contributes to real world dispersal and establishment. So what we did is we sequenced whole genomes from 160 specimens of Boletus edulis. From that, we identified these variants, these genomic variants that across the genome, and there's 790,000 of them. That's quite a lot. And this is a busy figure. What I want to highlight here is that when we looked for mixtures between the lineages, so this is a tree that shows you the lineages. We've color-coded them. Those lineages are geographically structured, very strongly geographically structured. And what this figure on the far right is showing you is just how much of the genome of the other lineages occurs in individuals of that population. So for example, in Alaska, you can see that most of it is an Alaska genotype, which is the pink. But this individual has a little bit of the EC genotype, which is East Coast of North America, and a little bit of some other genotypes. But by and large, most of these geographic populations don't show a lot of what we call admixture from the other populations. This map is showing the same thing. So these are pie charts that show the composition of the genotypes of individuals and their locations on the map. Now, there's a couple of things that we found that were kind of interesting that I'll point out. One is that we have a population that's segregating from British Columbia, and there's one example on the coast of Alaska. This appears to be a primarily coastal population, and we're trying to understand better how it's distinguished from the rest of the species on the West Coast. Alaska ends up being distinct from everybody else, and it also shows the most admixture. And we think that's because it's a zone of either current populations encountering each other again after being separated for a long time, or it represents an ancestral population, and all of these other ones have descended from it. We're still working this out. You can see the East Coast population. This is everything east of the Rockies. That's all its own distinct thing. This yellow one here, which is a European genotype, that was on a non-native Norway spruce. So we think that that's an introduction from Europe. We're almost certain of that. Okay, so one other thing I wanted to show you, and this is a little busy, but we were able to estimate the ages of these lineages. And they're actually quite a bit older than any of us anticipated. So you can see the youngest split, which is between Alaska and the East Coast, we estimated at about 1.5 million years old. So it turns out that these groups have been separated for quite a while, much longer than I thought. Okay, so I'm done talking about the Boletaceae, and I'm going to switch gears now, and I'll go through this rather quickly because I don't have a lot to talk about, and that's thanks to COVID. So it turns out that we got a grant funded to support this work, and we got one field trip in, and then COVID hit, and it shut us down completely. So we don't have a lot of new data, unfortunately. But to introduce this is best illustrated by leafcutter ants. So these are ants that cut fresh pieces of leaf material. They carry them into a subterranean nest, and they use that to grow a fungus. So the fungus is inoculated on the leaf, it decomposes the leaf, and then the ants eat the fungus, and that's their sole source of food. Now, a specialized parasite in the genus Escovopsis, this is a mold related to Trichoderma, has evolved in the system. And this is only known from the gardens of these of these fungus-growing ants. To suppress the growth of this mold, the ants have evolved a biocontrol defense, and that is pictured here. This is Pseudonocardia bacteria that the ants carry on their exoskeletons. That's the white that you can see. Those bacteria are producing compounds that actively directly suppress the growth of the Escovopsis. So this has become a model system to understand the origin and maintenance of coevolution. Yet there's still a lot we don't know about it. So there's a single origin of this, we know. It's diversified since its origin into about 245 described species in 19 genera. We know there are more out there. These are the ants. And the ants cultivate 4 distinct clades of fungi, which are depicted by the colors in this figure here. The mutualism's been around for a very long time. So it's been estimated to be 60 to 65 million years old. And this is partly based on fossil evidence. So it's a pretty reliable estimate. And interestingly, the fungus that the ants culture, they are transmitted vertically from nest to nest. And that's because when foundress queens leave the nest to make a new nest, they carry a bit of the fungus with them in a special pocket in their mouth. This leads to, over time, a process of domestication where the fungus will, because it's not exchanging genes with free living taxa, will evolve specialized features, very much like our own crop plants. Interestingly, about 20 to 27 million years ago, one group of ants switched to a completely different fungus. These are ants in the genus Apterostigma, and they started culturing a fungus in the Pteriolaceae family. Now, the Pteriolaceae is a diverse family of mostly hair-like coral mushrooms that are primarily found in subtropical and tropical areas. And they take a variety of forms, as you can see illustrated here. What we tried to do was understand better what might be happening in the fungi that are cultivated from this family. And in order to do that, we set out to, of course, sequence some genomes and start to look into the genome, differences in the genomes in the cultivated fungi versus the non-cultivated fungi, in hopes of finding the genetic basis of domestication. So this is just a phylogeny based on whole genomes. And the one thing that I wanted to point out from this is that it turns out that 2 lineages are cultivated by these ants, and they're actually not each other's closest relatives. So this suggests that there was either an ancestral cultivar that has had multiple escapes to a free-living lifestyle since it was first adopted by the ants, or the ants were cultivating more than one species and they have now sorted amongst the 2 groups of ants, or there were 2 domestication events. And we can't really distinguish between those 3 scenarios at this point, except one thing that might support the 2 domestication hypothesis is that the time at which they were domesticated, that is the time that they diverged from their free-living counterparts, are different. So one of them was 20 to 27 million years ago, but then the other one was adopted 17 and a half to 13 million years ago. So there was a difference in the timing of these 2 species. Okay. So one of the things we really want to know in the system is what chemistry might be mediating these interactions. We think that chemical ecology is probably a major feature of this mutualism in its maintenance. But we don't know a lot about it yet. So it's mostly a big black box. However, as I mentioned, as I alluded to before, we do know something about chemistry and its interactions in the system. And the example here is the chemicals that the bacteria on the ants' exoskeletons are making to suppress the growth of Escovopsis. And this is a molecule that was discovered from those bacteria. It's called Dentigeromycin, has no relation to me. It's missing an N, so it's not my name. And they were able to show with bioassays that this chemical specifically suppressed the growth of the Escovopsis mold. But what other chemicals might be mediating the other interactions, say the interaction between the cultivar and its ant host, or the cultivar and the mold? Again, we don't know anything about this yet. So we can look into what's known about some chemistry in the Terriolaceae, perhaps, to get some idea of what might be present. And there's very little known, not surprisingly, but here are some examples of chemicals that have been identified in Terriolaceae previously that do have bioactivity. So we do know they're capable of producing bioactive compounds. Whether or not the Terriolaceae that the ants are cultivating do this remains to be seen. So one thing we did is we started to look at the genetic capacity of the cultivar fungi relative to their free living, their wild relatives, to see if there was a change at all in the chemistry that they're capable of. And it turns out that there's possibly a pattern here, which is in the two cultivar lineages, you can see that there's a reduction in both the quantity and diversity of the genes that are involved in producing secondary metabolites, putative defense compounds. Now, interestingly, this is a pattern that you also see in domesticated crop plants. So domesticated crop plants tend to become more palatable, less toxic through the domestication process. And here we're seeing a pattern that's consistent with that. So this is really intriguing, although it's very, very preliminary. Again, so looking at the enzymatic capacity, so do these domesticated fungi, are they able to degrade the substrates that the ants are giving them better? You might expect that in a domesticated crop. Well, here we see, like in the secondary metabolite genes, a reduction overall in the number of genes that are thought to be involved in decomposition, in carbohydrate decomposition. So here, this might just be with the pattern in the secondary metabolites, might just indicate that there's been overall a reduction in the genetic diversity within the domesticated lineages, which is something that we've seen in other co-evolutionary systems. So again, there's some intriguing patterns, preliminary intriguing patterns that we're hoping to follow up on. Okay, so I'm nearly the end here. I just wanted to share with you a little bit about how we go about doing this work. So in order to do all that stuff I showed you, we actually need to go and collect these things. And we look in rotten logs here for gardens that might be hanging from the ceiling of the rotten log. They can grow on the sides of trees. Here, my colleague, Nicole Gerardo, is pointing a nest that is under the foliage here. You can't see it, but here's another one on the same trunk lower down. Sometimes they're in the ground, and we can find characteristic columns or holes in the ground that we know indicates that there's a garden of a fungus growing ant. And so we dig it out with a shovel. So this is an example of one that we had to dig out. And then when we do that, we collect that in the field. So here we bring spoons and we spoon the fungus garden and all of the ants that we can into a plastic container so that we can carry it back to the lab. We also collect free living relatives because we're interested in reconstructing accurately the evolutionary origins of the cultivar lineages. So we're also looking for these free living counterparts. And then we have to maintain these colonies. These are all living ant colonies that we maintained for a few weeks while we were collecting in Costa Rica. So they require a lot of maintenance, daily maintenance to clean them up and make sure they're hydrated and that kind of thing. And then when we get them back to the lab, we can try to isolate the fungus on sterile media so we can have single cultures of the different members of the system. And then we also maintain the colonies themselves, which really just requires providing the ants with a source of substrate that they can grow their fungus on and some water, and then they kind of take care of the rest. So we can maintain them in small plastic containers. And then that allows us to do stuff with them, like analyze their chemistry or do genomics or transcriptomics, that kind of thing. Okay, so we're really trying to take an integrative approach to understand a lot of complexity in the system. So we're trying to combine genomics with a bunch of other metaomics approaches, including looking at metabolites, transcriptomes, and putting it all together to try to understand the system better. Okay, with that, hopefully I didn't run too much over time. I would just like to thank some people that have been really critical in this work. My graduate students, which are pictured on the left. There's 2 current NSF grants that have supported these projects, so I'm very grateful to NSF for that. And then the picture that you see in the middle is an epic Porcini collecting trip that I made with my friend last summer in southern Utah. And this is the best collecting of Porcini I have ever had in my life. And we estimated we collected about 40 pounds in about 3 hours, and it was just endless. Everywhere we stopped, it was absolutely everywhere. So that was an amazing day. And hopefully you all can have those days in your future. So thank you for listening, and I'd love to answer any questions.
Peter, do you think you learned what your Minnesota bolete is?
No.
No.
I suspected that I'd lined them up with Verapes, at least one of them. But there's I'm confused about another one that about several other ones. Now the one of the ones that I know, or I think, we have growing in, conifers here is the Cariolensis, or I don't know if I'm pronouncing that correctly.
Yeah, so as far as I am aware, there are 3 species of Porcini in Minnesota. There's what I was calling Boletus nobilissimus, but turns out to be Boletus variopes. Then there's Boletus edulis, and then there's Boletus subceriolescens. I have not seen anything yet that is not those 3 species. I would say the one that's associated with the oaks around the Twin Cities and further west, the one that we were calling Boletus nobilissimus, is very variable, but everything I've ever seen is actually the one species, Variopes, aptly named, I guess.
Yeah, now I was listening to a podcast of you talking about the same subject, and you said you hadn't taken and sequenced anything that in deciduous forests in North America that was true Boletus edulis.
Yeah. So all of the records I've ever seen of Boletus edulis from broadleaf trees have been either misidentifications or there was a conifer present. So it's been a real struggle for us, actually, because one of the things we were really interested in with looking at the genomics of Boletus edulis was whether or not there were patterns of host specialization. But without enough records of Boletus edulis on non-conifer hosts in North America, we're not able to do that. So we have one specimen from, I think it's Oregon, that was found in a pure stand of populus, cottonwood. So that's one record we know of for sure. And then we have another one from an isolated betula birch tree. It was in the middle of a field with a birch tree. So that's also a good indication that it's associated with that host. But other than that, everything else we have comes from conifer hosts, or at least they come from habitats where there are conifers close enough that we couldn't rule them out as the main host. And so that's been really difficult for us. We have some anecdotal information that in northern Alberta, there is a lot of porcini in pure aspen. So we are planning a field trip there this summer to try to find some of those.
Cool. There are a lot of questions in the chat, which I can start reading next, but I'm going to let, will you say your name? Is it Payne? Payne? Sorry.
That's okay, I get that all the time. It's Fane.
Payne, perfect.
I just wanted to say, Bryn, it's a pleasure to meet you, and it was awesome to listen to your presentation tonight. I am really glad that I took the time and found out about this and sat in on this. That was amazing information you were giving out. Some of it a little above my head, like the DNA sequencing and stuff, but the information that you brought forth and that you're finding in your research is just like everything else with mushrooms. It's mind-blowing. It is epic. I love it. The more I look into this, the deeper the rabbit hole gets. And now that I'm finding out that the deeper the doctors and the scientists look into this, the deeper the rabbit hole gets for you guys too. So just thank you. You did a great job on the presentation. The slides were awesome. The whole thing. I mean, I give you a 10 out of 10, buddy. And just thank you a lot. And the ant farming one is just amazing. I love all the different symbiotic relationships they're finding between fungi and mycelium. And it doesn't matter what species is with, humans to ants. It's amazing. So wanted to thank you for that, man. It was really great. And I look forward to looking a little more into your research and hopefully being able to get a copy of the meeting from somebody tonight who recorded it to watch some of this again. That was great. And thanks, man. You have a good night.
Great. Thank you very much.
Yeah. So
Good.
Bryn did give us permission to record this, so it will be on the MMS YouTube channel. And the first question in the chat is, do you have a YouTube channel?
I do not. I'm sorry.
In your spare time. But he was featured on the podcast, Welcome to the Mushroom Hour, which a lot of you, if you haven't listened to that, that's like an impressive variety of topics as well. The next question was, what do you find interesting about Vietnam? Is there a specific region in Vietnam where we can find interesting mushrooms, maybe porcini?
Yeah, everywhere in Vietnam, actually. I only had the privilege of collecting in 3 places in Vietnam. I was in Da Lat, which is in south central Vietnam, and it's higher elevation. There's pine. It's all pine forest, and then lower down there's some Fagaceae oak family dominated forest that I collected in, and then I collected in lowland dipterocarp forest at Cat Tien National Park. And then also on the coast in an old littoral forest. So this is old beach that's been colonized by these dipterocarps. And that was absolutely astounding collecting. Something about sandy soils, and some of you in Minnesota might know this from going to places like Alice in Savannah or something. There's something about sandy soils where it just can be so productive. For mushrooms when you hit it right. It's just something incredible. And I just happened to hit this habitat right in Vietnam. So that was fantastic. I'd love to go back. I think Vietnam, like its other biota, probably represents a crossroads of stuff from stuff that you find in present day Northern Asia and stuff that you find throughout Southeast Asia and the archipelago going to Australia. And so for that reason, I think it's really biogeographically interesting.
Very cool. The next question was, do you have any recommended reading?
Just general reading. I really enjoyed Entangled Life. I don't know if anybody else has read that book, but I thought that was an excellent book. Yeah, I don't know. I don't have much time to read these days. So.
Awesome. The next question is, any evidence that the leaves being fed to the fungus by the ants are also being subjected to a domestication process? Are they doing anything to farm the leaves or change them?
No. So the leafcutter ants are, they're actually the dominant herbivores in the American tropics. And they utilize just about everything they can. But they will change what they harvest depending on feedback that they get from the fungus, actually. So the fungus is very sensitive to changes in carbon and nitrogen ratios. And there is some signaling that's going on so that they can start, the ants can start foraging for things to get the optimal ratio of carbon and nitrogen in the substrate. They also, a lot of plants have toxic chemicals. And so the ants, well, actually the fungus appears to have undergone some evolutionary changes that makes it much more capable at detoxifying a lot of those compounds than the free living counterparts can. So that might be one of the innovations that has made that system so successful. But the ants also will change what they forage to ensure that there's not too much of one thing.
That's so crazy. It's like, yeah, just mind blowing. Another question, are there any new approaches to preserving mushrooms, mushroom specimens in Fungaria?
Well, I mean, drying is still the tried and true method for preserving specimens. We use silica gel to dry them when we're in the field in the tropics. Anecdotally, I would say that silica gel seems to be less effective at higher latitudes, and I'm not sure why that is. So heat drying seems to be best still, up, in Minnesota or Utah, generally speaking. But we also now are really, we're much more aware of the need for intentionally preserving tissues for DNA and RNA. And so we now regularly will flash freeze tissue and put them in an ultra cold freezer. That preserves the RNA so that if we want to go back and look at that, that's available to us. So that's not something that is traditionally a part of a fungarium. But I think people like me, because we primarily work with DNA and RNA, we think a lot about that stuff now when we make collections.
Perfect. Yeah. You have to get one of those fancy freeze dryers that a lot of the
yeah, we have one of those actually. Yeah.
Nice. Another question, is it pure coincidence that the unique bolete type, it says colored green circles on the slide, is in the same geographic region as one Mr. Paul Stamets and his crew?
I think that's purely coincidence.
And then Ron was asking, do ants farm Lepiota species that you know?
Yeah. Hi, Ron. It's been a few years. Let's see. Yes. So the problem is that the taxonomy of the group of mushrooms that most of the ants cultivate, is really poorly developed. So it's the Leucoagaricus Lepiota group. And so we still haven't sorted that all out. Hopefully we'll get there eventually. And there's been a lot of progress made recently that should be coming out fairly soon. So hopefully that'll get sorted. But they're probably not true Lepiotas. They would probably be best classified as Leucoagaricus.
Interesting. And then do you still collect Porcini samples? And if yes, how would we send them to you?
Yes, we are still collecting Porcini. We love, especially Boletus edulis. We told NSF we were going to sequence 600 genomes of Boletus edulis, and we've only done 250 or so. So we've got a little ways to go. So yes, we would love more Boletus edulis samples. And I can send you my address. I can share my address to send them to.
Great.
Somehow.
We can look on your lab website too, but if it's different or if it's more specific, yeah, please let us know. I'll email you after, and it's something we could share with the club.
Great.
Yeah, it'd be fascinating to hear more about your research in future years as well. What else? Oh, okay. Which book, website, or paper provides the most up-to-date keys for Porcini?
Well, they're already out of date, so you're gonna have to wait on that one, I'm afraid. We'll try to get to it this year.
How do mycologists determine the age of an evolutionary divergence?
Yeah, so ideally you would be able to use fossils and place them with confidence in an evolutionary tree, and that would give you a calibration point that you could use. But generally speaking, we don't have that. There's not a lot of fossil evidence for fungi. They don't preserve well. And even if they did, or in the few cases that they have, it's really hard to tell where they belong in the tree because the morphological features that we have to look at just are not going to allow us to do that with much confidence. So mostly people use, they use a molecular clock estimate. So we know that on average, DNA mutations will accumulate at a certain rate. And so we can use that rate to back calculate the ages. So that might sound like a lot of hand waving, and it is, but we make efforts to try to rule out or put some confidence intervals on those dates. In fact, if you recall, the figure that I showed had these big long red lines. That was the confidence estimate for the date that we could estimate. So yeah, hopefully that answered your question.
And if anyone wants to unmute when I'm getting to your question in the chat, please feel free. Hannah Miller, you have a great question about the role of art in taxonomy. I don't know if you're. She was asking, I was intrigued by your mention of species being identified with paintings only reference. Yeah, I have a few of those books where, yeah, there's like the beautiful watercolors of the specimens.
Yeah, so I was referring to the 4 European species, Boletus aerius, Boletus reticulatus, Boletus edulis, and Boletus pinophyllus. And those were all described way back when and sanctioned by Elias Fries. And he based them on paintings that were found in books by Pierre Bouillard and some other people, not remembering him right now. And so because those represent the types of those names, they're now known as iconotypes. And a lot of the old European names, that's what exists to represent them, are paintings only.
Interesting. So even what you guys are meaning is even when someone hadn't collected and studied and specifically documented them, but what was found in art that give us clues even. That's fascinating.
Yeah.
Wow.
Making
Yeah,
specimens and keeping them wasn't a common practice in the early 19th century. So
this is not the best example, but I even find that when you're in the woods, sometimes you take pictures of the things that you don't know what they are or that you want to study more. And if I was to look through all the photos I have to find a picture of morels, I really don't have any because I'm like, you just pick it and eat it. And it's like, oh, I know what that is. So, you know what I mean? You sort of, there's this bit of like, I know that check versus I don't know that I better document it. And it kind of makes you pass up a bunch of stuff that you've kind of checked off as knowing that maybe you don't because you haven't really studied it close enough. So yeah, I feel like your lecture is scratching the surface on so many things that our heads are just spinning with all these different things that are possible. So any other questions? Oh,
I have a question.
Great. Yes.
So I wonder during winter, does the Porcini mycelium survive? And at what temperature do they become active again?
I can answer the first part of that, which is yes, they persist. They are perennial organisms. We don't know how old an individual can get, but anecdotally, they can get quite old, probably as old as their hosts. But the second part of the question, I don't know the answer to. Nobody's looked at that as far as I'm aware, but probably it'd have to be above freezing.
Thank you.
Yeah.
Now, if you were going to look for a Boletus edulis in Minnesota, the true Boletus edulis, where would you look?
I always found it up north, but I mean, this is this is ascertainment bias at its best.
Sure.
I'm from Duluth, so I spent most of my time looking around Duluth. In fact, in Duluth is a great place for it, but I would say I most consistently find it near balsam fir. So if I were to target a particular habitat, that's what I would spend my time looking in.
So balsam fir, and those are Boletus edulis that have the same kind of morphology that you kind of see in the guides.
Yeah, in Minnesota, the variety that we have, variety in loose sense, is paler. The cap is paler and tends more towards the yellow to orange colors than the brown that you would see, say, out west.
There's another question about the variety of tastes for cooking in the genus with so many different species.
Well, I haven't tasted many of them, so I don't really know. Some of them are nuttier than others. So Nobilissimus, Variopes is nuttier in flavor, I think, much stronger, nuttier flavor than true Edulis is, at least in Minnesota. So yeah, but other ones, there's a really divergent group. So there's kind of two major clades within Porcini, and the other clade, which includes Boletus separans, that's the one that people know best, although I don't think it occurs in Minnesota. I've never seen it. That tends to have an acidic flavor that in age can be quite rancid. They can smell kind of nasty. So yeah.
Interesting. Scott Benjamin is also asking, besides balsam, if you were going to plant a conifer to encourage boletes up north, what would you plant?
If it's Edulis, I think spruce and balsam are the two major hosts.
What Edulis clad or related species boletes in Minnesota, are you aware of that you've identified growing here?
So other Boletaceae?
Yeah, I mean, you said Edulis, you said Varipes. We talked about subcerulescence.
Those are the only three Porcini that I know about. I'd love, if somebody thinks they've found something other than those three, I'd love to know about it for sure. But those are the only three that I've ever seen there. But there's lots of other Boletaceae, of course.
Yeah. I mean, I've found Porcini for many years. I find Porcini that at least morphologically they appear separate. One of them appears to be Varipes. Another one, I don't know what it is. I've seen it compared to Atkinsoni. And another one is now being compared to Cherokeeensis. And but they have very different appearances.
Yeah, I suspect those are all varieties.
Okay.
But I'd be very happy to sequence them. If you want to send me what you think are those three, I would love to.
We can arrange for that.
Okay. I only need a little bit.
Well,
Peter, did you mean Chippewaensis?
Or Chippewaensis.
Yeah, that's what I thought you meant.
I'm sorry,
Yes.
Chippewaensis.
Yeah, thanks.
I'm sorry.
Chippewaensis is just an Edulis. Yeah, we've sequenced the type and it's just Edulis. So it's just one of those morphs.
Okay, so is it considered really a separate species then, or?
No, not by me.
Okay. All right.
Yeah. So great.
Except I find the ones that I'm seeing that look like that, I find in deciduous forests.
Oh, interesting. Like aspen or what?
Oak.
Oak. Oh, yeah. Please send me some. I'd be very interested to have those.
Yep.
Perfect. Great. Well, I don't see any more questions in the chat. I just really, really loved this conversation.
Very much so.
Presentation, and yeah. So, like I said, our minds are spinning. So thank you so much.
My brain is going to explode.
Perfect. And thank you, everyone from the U of M who joined as well tonight. And yeah, it's the perfect thing to do on a cold Minnesota night is dream about finding boletes. I'm learning a ton more about them. I know when COVID is over, or if things open up, you'll probably take the ant research a lot further, and we'd love to have you back anytime you're interested. So,
Great, thanks. Yeah, we're finally making a trip to Panama in May, so that's
oh
exciting.
wow,
Yeah.
great, awesome.
If you're not totally taking your time off in Minnesota away from mushrooms, feel free to keep in touch.
Great, thank you.
Yeah, awesome.
Thanks,
Yeah,
Bryn.
if you're in Minnesota visiting family and want to go out hunting, I'm sure someone could show some of their spots, especially Peter. You have a lot of spots.
I try.
Sounds great. I miss the chanterelles. I can't find many of those out here. So,
Yep. Well,
Great.
There are some here.
Well, the chat is just filled with thanks and
Very much.
praise
Thank
for
you.
just so many thanks. All right. Well, everyone have a wonderful night. It's great to see everyone's faces too. I until we can be in person again, I really enjoy seeing people on the Zoom.